maybe some of you know, I only recently moved to Cornell. I was a faculty over at Yale for 9 years, and then I moved here in July. I thought I'd give you just a view first of my lab. This is the people in my lab that actually does all the work. So on the top left is a photo we took actually back in New Haven. I think majority of the lab, maybe 2/3 of the lab moved over to Ithaca with the lab in the summer. So we have 4 postdocs, 5 PhD students, and then 1 postdoc right now in the lab. Carly is an animal technician, and then Farid is a master's student. So there's about 10 full-time people in the lab. In terms of the research, I thought I'd first give you just a bird's eye view and what we're interested in. We as a lab, we're very fascinated by drugs and psychoactive molecules. So we're very curious how these small molecules can cross the blood-brain barrier into the brain and then does something to your brain structure and brain function, and then eventually lead to some fairly profound behavioral changes or desirable behavioral changes. And one of our goals for studying drug action is that we want to be able to understand and predict then what some new drugs might do to your brain, and then in order to also predict what their behavioral effects might be. And you can imagine this is quite important for drug discovery, drug evaluation, and just generally pharmaceutical development. So first, I'll tell you a little bit about the methods that we use to measure drug action in the brain, and then I'll go into some specific research that we're doing right now in order to study subclasses of drugs. And I should add too, feel free to ask any questions in the middle. I mean, I hope this will be interactive, and yeah, I think that will also make the presentation go well. So I know I'm just going to go start from quite basic, right? So I know not everybody is a neuroscientist here. So if you look into your brain, there's 86 billion brain cells in each of our brain, roughly speaking. And then this is one like prototypical brain cell or neuron and how it look like. You have a body of the cell in the middle there. And then what's interesting about brain cell is it has very elaborate processes for communication. So there are the so-called dendrites up in the top that receive input. And then there are axons on the bottom that receives output. And we're very interested in what drugs do to these different compartments within the brain cell. In particular, there's a lot of evidence, and I'll show you too from my own lab and other people, suggesting that these drugs can particularly impact the dendrites, which is where the neuron receive input. And this has a good, strong implication on how it affects neuron communication. And that's how we think many of these drugs primarily act. So to study how the drug act, and we use animal models. So we use mice as an animal model, and we do optical imaging. And then we're imaging a frontal area of the brain. This is homologous to the human prefrontal cortex, which is where a lot of executive function and cognitions are being managed. And then we use viruses or transgenic animals in order to label specific neurons so we can image them. So here, the third panel shows an example where we injected viruses to introduce fluorescent protein into the brain so then we can later on image a fluorescent signal and see where the neurons are and what they're doing. This is how the microscope looks like if you won't join the lab tour. But on the left is one microscope that we have. These are so-called two-photon microscope. Actually, a number of BME faculty member, Chris Schaefer, Nozomi Nishimura, they also have some of these very similar microscope. Chris Su is also a big developer of these microscopes over at Applied Engineering Physics. So this is a very good community for doing this imaging. But this is one of the 3 that we have right now in the lab. And you can see on the left is the objective coming in to do the imaging. On the right is sort of the bulk of the microscope. And then the video that was played there on the right was a mouse that's being imaged under these microscopes. And you can see that it was running a little bit on the ball. So they're awake, but they're fairly comfortable. They're fairly habituated and quite used to this imaging setup. Yes, question? Yeah, so the question was, how is the strength of the Cornell community in optics maybe help this research and attract the move when I initially came here?
And the actual physical infrastructure.
Yeah, so I think that was one of the, I would say, the top 2 reasons that contributed to this. As you see, many of the research that we do involve this type of imaging. And we're at a point, I think, where we're using the best of the current technology to image these drug action. And then what Cornell offers is that we can maybe push that boundary even further because the number of groups here that we mentioned, again, Chris Schaefer, Nozomi and them, and me myself as well, we're trying to develop new methods. And I think there's a core community here where we can really push that boundary by applying some of these engineering principles to develop new imaging methods that will be very suitable for studying drug action. So that's something I'm extremely excited about. So these are kind of images that we can get with the two-photon microscope in a mouse brain. On the left, again, is the schematic of the neuron, the cell body, and the dendrite. On the right is the imaging and then reconstruction of a single neuron that we have labeled now in the mouse brain. I always like watching these videos. I think it's very beautiful. The dendritic tree is just very striking, how long it is. I think, it just separates these cells, how it devotes so much material to communication. Most people, I think, in the neuroscience, they care about the cell body, but I want to emphasize, again, much of it is actually about the communication and the processes. You can see there's a main trunk that come out, the apical dendrite, and then it kind of fan out, and then eventually on the top is when it actually really receive a lot of these long-range input in the brain. That's when it really have a lot of so-called apical tuft up there. Not only can we image the structurally what's going on in the brain, but we can also image dynamical signals. So here, instead of loading a fluorescent protein that statically give a signal, we can also put in a sensor that changes the fluorescent signal whenever there's calcium ion going into the cell. So what you see here, whenever the cell is flashing, it means there's an influx of calcium going in there, which means when the neurons are active. So you can also get a dynamic view, like a live view of the neural activity in the brain using these kind of imaging methods. Question. That's a good question. So the question was, are all the drugs that we're testing mental health, and then how do we think about, I think, kind of brain and peripheral interaction? So I will get, after I go through some of the methods in about 3 slides, I think we'll start going into the drugs. I'll talk more about that. But to get at that point, typically drugs these days are administered either orally or it could be through infusion. Both of those go through the peripheral system. You're not directly injecting in the brain. So there is interaction just because of the route of entry. Some of the drug can be targeted, so then you mostly have your desired effect in the central nervous system. But particularly for the drug I'll talk about, for example, like the psychedelics, they act on serotonin receptors. And it turns out there's also a lot of them in the heart that's not very good for you. And it damages your heart valve. So those are definitely considerations. It's not something that we study per se because we don't look at the heart, but it's something we think about. Just one final, I think, example of the methodology. We can also not only look at the cell body activity now, we can also move our imaging plane of the microscope up to the top, to the dendrite, and also look at these dynamical signals up at the dendrite. And this is also a movie that always struck me as well, how dynamic it is, like different dendritic branches come in, signifying when the neuron receives a lot of inputs from other cells. Okay, so that tells you briefly how we image the neurons in a mouse brain, and then now I'll talk about some of the drug that we study, and then also what my research have produced and some insight that we've generated. So the primary class of drugs that my lab is interested in are antidepressants. So these are drugs that help for treating depression. So depression is obviously a very devastating mental illnesses and affect a lot of people. Particularly relevant at Cornell, I think. A lot of the student body also have some of these symptoms. And I think current treatment are quite lacking. So I think a primary first-line treatment, the easiest thing is to get drugs, and a lot of people are in fact on antidepressants, but they're often not very effective at least for some fraction of the people. They're not responsive. Some people are responsive. And then also usually now it takes a long time to work, several weeks in general. And then there are not a whole lot of other alternative treatments. So you can have psychotherapy, which is very time intensive, and then you can have electroconvulsive therapy, which is quite aggressive. It's just not a whole lot of treatment, and a lot of those also kind of came from decades ago. So there's not really a lot of new innovation in this area, which is why I think research is really urgently needed. So my lab are quite interested in newer generations of antidepressants that people think are could be quite efficacious. In particular, some of the molecules that we study are ketamine, which has been approved 2 to 3 years ago by FDA as a new class of antidepressant. So that, along with brexanolone, is turned to 2 newer approved antidepressants recently. And then another one that's not approved right now, but there's a lot of study, is the use of psychedelics for treating depression. So that's shown here. So on the left is the compound psilocybin. And then in the body, if you take psilocybin, it rapidly metabolizes into psilocin, which is the molecule in the middle. And then that's what gets into the brain and bind to the brain receptor that exert the behavioral effect. And so you might know psilocybin because it's the active ingredient in the so-called magic mushroom on the right. This is a photo of one of about 200, I believe, species of the psilocybe mushroom that contain these compounds. And you might know, magic mushroom and the effects that they have. I mean, just through popular media. But here I'll also show you one clinical report of a person, a woman, adult, who has taken, who had been administered psilocybin, and then afterwards they asked her what the experience was. And then on the left is a drawing that she made of that experience. So you can see, and I'll read it out aloud. I can't see my computer come out here. My inner vision became the universe, filled with colorful waterfalls, glistening like stars not bound by gravity. They floated in the air and folded around each other, totally quiet. The streams were infinite and beautiful. So I picked this passage because it highlights several aspects of the experience. Some people call it the psychedelic trip. Right? These are acute experiences that people would receive within, typically for psilocybin, 3 to 6 hours. This is when the height of experience. You can see that it changes the visual perception, which is a very common feature of a psychedelic like psilocybin. And then there's also a sense of connectedness with the universe, or other people call it the mystical-like experience that one might receive as well. So these are the acute effects within the 3 to 6 hour time point after consuming these compounds. What's quite exciting for the field of psychiatry, though, is that after the acute effect has worn off, there seems to be also longer-term effects on the days that follows. So there are now a number of clinical trials. These are phase 2 clinical trials. So with people, a population size, sample size of roughly between 50 to 200 people, depending on the trial, done at Imperial College London, Johns Hopkins, and also by a company called Compass Pathway, that show that if you administer psilocybin and either compare with low-dose psilocybin or placebo, you can see a marked effect on depressive symptom in patients. So on the left here, the plot, you can see the depression score, which is how clinician would characterize the severity of depression symptom. You can see the drop. And perhaps the most striking effect you can see is when you compare it to the conventional SSRI. So for example, Prozac, which is fluoxetine, you can see that at least in these early trial, it seems like the effect is much quicker, which I think is quite important because particularly depression sometimes come with suicidal thoughts, so you really want a drug to act quickly when you need it. And then it's also quite sustained, the effect, compared to the SSRI. You have to remember the fluoxetine is something you have to take every day. And medical adherence is difficult, so a lot of people would stop taking the drug at some point. Whereas if it's really, these early trial can be replicated, you can see for the psilocybin case is 2 doses. So the effect is much more sustained with a few doses. So that's why I think there's a lot of excitement and promises for these compounds for treatment of depression. So as a result, there's also a lot of hype. I think that's a problem in this area right now. I think when I started studying psychedelic and their effects on the brain 3 years ago, there's not a whole lot of press, but now there's just a lot of excitement and a lot of writing about it. I think it's kind of, the pendulum has swung almost off to the other side, right? And there's a lot of also unsubstantiated claims also as well. I think it's also being thought to treat many other things where the evidence is weaker. So I think there's good evidence for things like depression, cluster headache, substance use disorder, but many of the other diseases and ailments I think are a lot less proven, but doesn't stop people from writing about it, which is a problem. So some of the work, I mean, the ones I show I don't think are exaggerated, but I'll give some example on what the coverage is like. a lot of this renewal interest in psychedelics started from this book from Michael Pollan, How to Change Your Mind. Maybe some of you have read it. It's a bestseller. TV shows, Fantastic Fungi. I think How to Change Your Mind also now on Netflix. Hamilton's Pharmacopoeia, which is actually a very interesting series on drugs if you're interested in this sort of thing. There's now also Popular Press. Newsweek has a cover. New York Times talk about it. And then finally, if you follow the British royalties last week, 2 weeks ago, Prince Harry talked about how he uses psychedelics to deal with grief, which I think is really not kosher, right? So I think there's an opinion on New York Times, I think, saying how you should not do that. Yes. But it is also quite an exciting time to do research on it. So I was at the Neuroscience annual meeting 2 months ago. We have a symposium talking about the basic science behind these drugs, and it was packed house on the photo on the top left. It was fun also to talk about these things with the press and popular media. So I'm a big fan of NPR, so I was very excited to get on NPR and can talk about these things. Also, the Connecticut Public Radio, I listen to this every morning when I drive in. It was very fun to talk about some of the research on these shows. And I think for academia, it's also quite exciting. There are a lot of philanthropy in this area. So Johns Hopkins was one of the first. I think they have a $90 million Center for Psychedelic. UC Berkeley received also, I think, around $10 million. NYU, Mass General, Imperial College London, and then University of California, San Francisco. So there's a lot of research center that's popping up, fueled by, I think, interest in this area. Again, for the potential that how it can transform psychiatry. So, okay, I talk about how we measure, and then I talk about why it's important to study some of these compounds. Now I'll give you a few examples. We won't go too deeply into it. Just a few examples on what research my lab has done and how that has helped our understanding of what these compounds do. So this is work done by graduate student Clara Leal and then also postbac Ian Gregg. So one of the first things that we want to know is if you give a psychedelic to a human, you know that they are tripping, you can ask them. But if you give them to a mouse, like how do you know it does anything? You can't ask the mouse and see how they feel, right? So it turns out that if you give these compounds to a mouse, there's also a very immediate behavioral effects, and that is it causes the head to twitch. So here, the video you can see on the left is the mouse that received a psilocybin. Hopefully you can see that rapid head movement. This is actually, the video is slowed down 6 times. The head movement itself, it does it about 90 times a second, the frequency. It's actually really, really fast. On the right is a mouse that just received salt water, saline, so it doesn't do anything. So this tells us that the drug is psychoactive in an animal, and this is the right dose to study. And we can map the dose. So here, just give it different doses. You can see that as we increase the dose, the amount of head twitches increase as well. So for most of the study, we end up using that dose of 1 milligram per kilogram, which is when it starts to do these head twitch. So again, it's doing something in the brain. One of the study that we did is using the imaging method that I told you about imaging the structure of the dendrite. And we're very interested in seeing whether these drugs changes the morphology and the structure of the neurons in the brain, in the frontal cortex. The idea is that there's a lot of talk about these drugs being important for neuroplasticity, so it changes the structure of brain circuits, but there's actually not too much evidence of it happening. So this is one thing we want to investigate. So this is the timeline of an experiment. So we would do the surgery that I showed you with the glass window and all that, and then we would image it before, and then give the animal either the psilocybin or a saline, which is a vehicle, a control, and compare, and then image again 5 times following that. So this is the kind of image that we can get then. Again, using the same 2-photon imaging strategy to look at the neuronal structure. You can see the Ling Xiao here, the postdoc who's leading these experiments, done an extremely good job, right? You can track the individual segment over days and go back to the same segment and track it over days. You can see the individual, those knobs that coming out, the protrusion is coming out of the dendrite, which is where the connections are being made. The little thing that we call them the dendritic spines. And again, you can see them over days. Most of those dendritic spines are stable, so you don't want your brain to keep changing too much, right? You want to maintain your memory and your skills and so forth. But there are a subset that form over time and also some that get eliminated. So our brain is shifting a little bit over time. And we can track that with using imaging in the mice. Oh, they're moving around. So when we image it, we would have to fix the head just because these are, again, microscopic images that very detailed. You cannot have the head movement. But after the imaging, they just go back to the home cage and they do whatever they're doing there. Actually, one important point is also the drug you can see is administered, also not during the imaging time, just so not to interfere with that process. Yeah. I think we developed, Ling Chao developed some elaborate protocol. There are a lot of landmarks in the brain, like some blood vessels and so forth, that you can go back to the same place. So the question is an important one, which is in human, we're talking about its potential for treating depression. But in a mouse, how can we maybe translate some of these findings eventually towards the therapeutic effects of these drugs? Because obviously, mouse don't have depression. They're not humans. I think mental illness is quite unique to human, right? They just don't have the capability. So how I see this research fit in to that bigger picture is that in the mouse, we can have a very precise understanding of how these drugs work on brain circuits that are not the same as human, but enough similarity, a mammalian circuit with the same kind of cortex in the brain architecture, which will then eventually be quite helpful to serve as potential biomarkers for searching for new drugs that might be better, or more effective, or safer. So one issue is you can't, and I don't know if I'll talk about it later, with psychedelic, psilocybin is one of many. There are probably thousands of analogs. How do we begin to ask which analog is a better drug that we should test it in human? A lot of times, pharmaceutical company or researcher will have to find some lead compound and some ideas on how these drugs work to optimize it. So I think our research, through basic understanding of how these drugs work, can bring a lot of value to that process. Yeah, but I don't think, at the end we can understand why these drugs are therapeutic in a sense. I think that's a really human question, right? Whether they can treat depression. But again, we can provide understanding that can help to look for new compounds. The question was when psilocybin is administered, are inactive parts of the brain becoming active? So we have one follow-up study, which I'll not show here, where we try to look at gene expression changes across the whole brain. Yes, when a drug like that applies, because it's applied systemically, which means that it's applied, for example, oral. In the mouse case, we inject it, but then it goes into the whole brain, right? We don't limit where it goes in the brain. It does affect many places in the brain. So some places it goes up, some places it goes down, depending on the type of receptors that in that region, as well as the circuit architecture in that region. What we see is changes in the neuronal connectivity, but we haven't explicitly looked at the amount of brain cells. But usually in adult animals, the amount of neurogenesis is relatively low. So we were kind of born with a set of neurons, and then through adolescence, that actually there's a lot of pruning and lowering of the brain cell. And then over time, we also lose them, whether it's aging, alcohol, or other things. We don't grow them back, unfortunately. Yeah. And I don't think there hasn't been too much evidence of any drug that actually can grow new brain cells. So yeah, the main finding for this study that we published last year was that if you give the mouse a single dose of psilocybin and then look at the number of neural connection, or the proxy, which is those dendritic spines that we're imaging, you see that the single dose of drug leads to about 10% increase in the density, which is the number of the neuronal connection. And then you also see these dendritic spines got bigger, which means that they get stronger. There's also about 10% change. And then one of the interesting things was how long this thing lasts. So the change in the density actually lasts up to 34 days, a month later when we look there.
After one dose?
After one dose, yeah. So even though it doesn't produce new neuron, it does seem like it promotes the formation of new connections between neurons, which is quite exciting. And I should say also the reason why this is interesting to a lot of people is that the time scale of it. So a lot of the things you can measure the drug effect is quite acute, maybe correlated to that acute behavior effect. There's not a whole lot of things you can find in the brain after the drug administration that lasts for so long. So these changes in neural connection, the time scale seems to match the therapeutic time scale, and that's why there's also a lot of excitement about it. Maybe a trace of the drug's long-term action. So yeah, I already sort of maybe mentioned this a little bit, but the reason, again, why I think this may provide some insight is that there's a lot of suggestion that you have synapse losses associated with depression in these brain areas in humans. So the fact that you can reverse that and maybe add some neural connection to the brain could be helpful. And then the second thing is that some people also think that the actual acute experience is quite important. And then that itself and the integration of it could also be useful for reversing depression symptom. So by virtue of adding and promoting connection between neurons, that enhanced plasticity could also allow you to maybe remember or learn those experiences better. And then that could also eventually potentially be a mechanism for the therapeutic effects. Yes, so in fact, psilocybin, there are a number of trials now. I think there are either phase 1 or phase 2 to look at effects on PTSD. A more advanced clinical trial is with the drug MDMA, which is not exactly a classical psychedelic, but it's very related. It's called entactogen. It has some similar kind of effect, but also a more pro-social effect as well. You feel very social. And yes, it's definitely being tested on PTSD. Yeah. So I think the idea is quite exciting, right? To harness the brain's plasticity potential, which is what's already there. You're just trying to heighten it to let it rewire and correct what's wrong in there. You're not really trying to push it. So I think that's why I think these drugs could have a lot of potential as well. Okay, so just to give a sense on what we're working on right now. Right now at Cornell, Neil is a MD PhD student from Yale who moved to the lab. One of the things we're interested in is, okay, we see more neural connection, but how does that happen? So we think the calcium influx and that signaling is quite important. So that's one thing that Neil is doing right now, using some of those dynamic imaging approaches that I show you to image the changes of calcium signal within the dendrite. So this shows you one example. We're imaging the soma, which is a cell body, imaging a number of dendrites in the cell, give them psilocybin, and then just see what happens. And we hypothesize that the changes in these calcium signaling could contribute to the growth of the new neuronal connections. Another thing that we're doing right now is another MD student in the lab, Pasha Davodian. What he's doing is, I think actually that question about whether it changes neuronal activity in the brain. So to measure that, instead of doing imaging now, we're actually putting electrodes in there. So these are microfabricated electrodes, so they have tiny pads. They're fabricated in clean rooms, and we can measure thousands of neurons at once. So on the right is an example of the electrical signal that we can get. And we can ask that question on how it changes activity in different brain regions as a response to psychedelic image. Just to conclude, right, so we are excited about the possibility of helping to solve the problem of depression. And our approach is to hopefully improve the treatment in terms of the medication. So for that, we're investigating the basic mechanism of some of the most promising compounds. And we hope that some of those insights could allow us to accelerate drug discovery and drug evaluation. And I think one of the long-term goal, I hope for a future where depression is also not a homogeneous disorder. There are different subtypes of it, and there are constellation of symptoms. So one possibility is that by discovering new medication, you might start to have medication that would be suitable for certain subtypes. And then that would be allow for more precision type of treatment. So not necessarily to replace existing treatment, maybe SSRI, but some of these newer compounds might be particularly suitable for subtypes of depression, and it can be much more targeted. I have a couple, I guess I lie, I have a couple more slides, but just more about the lab. So that was the science. But I want to say that these are some of the principles in the lab that I share as well with the lab. So a motto of the lab is we want to be creative and be bold. I think we want to try new things in the lab. I want to teach the student how to do first-class research. And finally, I'm a big supporter of open science. So we post all the manuscript on preprint server to share, and then also we post all our code and data online. I think that's an important part. I mean, it used to be you publish a paper, it gets stuck in a library or something. But now, I mean, you have storage, right? You can share data and so forth. You don't necessarily limit everything to a piece of paper. and then other people can use and manipulate the data. The lab also support diversity and inclusiveness. That's an important part of the lab. And then finally, I want to say we also like to do outreach. So at least me personally, I'm involved with a couple initiatives. There's a pen pal project. I write letters with students. And then another thing I've been doing is the Skype a Scientist, which is always fun. You have a class of kids, and then you Skype. And then I like to bring the laptop around, and then you can see in the lab what's going on. And they get very excited. There's a mouse right there, right in front. So, yeah.